Hey everyone, and welcome to this explainer. Today we're doing something really cool. We are taking an exclusive behind the scenes tour into the laboratories of the future. We're going to explore the absolute cutting edge of modern biotechnology, where scientists are literally engineering superhero crops. It's a massive journey, going from the microscopic, super precise editing of DNA all the way up to global digital farming. So get ready, because what we're about to cover, it's going to completely change how you look at the plants right outside your window.
Okay, let's dive into this. Here's our roadmap for the tour. We're going to start at the molecular level with the biological photocopy machine. Then we'll scale up to building the super plant, step inside the magic plant laboratory, and finally wrap up with breeding the future. All right, first stop, Section 1, the biological photocopy machine. Imagine stepping into the lab of a molecular biologist, or as our source text playfully calls him, Professor DNA. This is where the fundamental editing machines of biology live and breathe. Now, to really grasp genetic engineering, you first got to understand gene cloning. Basically, it's the process of isolating a really useful gene from one organism, popping it into a vector, transferring it to a host cell, and just letting it multiply. You know, it's exactly like a biological photocopier machine. This single core principle, it's the whole reason we have life-saving breakthroughs today, like human insulin that's literally produced by bacteria. Crazy, right? Now, what's really interesting about this slide is how these highly complex molecular tools can actually be broken down into everyday metaphors. It makes them so much easier to remember. To cut the DNA at very specific sites, scientists use restriction enzymes. Think of these as our molecular scissors. To stick pieces back together, they use DNA ligase, which basically acts like biological glue. And to transport these brand new genetic instructions directly into a cell, they use vectors. You can just think of vectors as microscopic DNA delivery trucks. But not all delivery trucks are exactly the same. There's a pretty big difference between a cloning vector and an expression vector. A cloning vector's main job is just to copy and multiply DNA fragments. It's strictly a transport vehicle. An expression vector, on the other hand, is like a specialized, high-tech manufacturing truck. It comes equipped with extra features, like a promoter, which allows it to actually unpack its cargo and manufacture the protein right there at its destination. Moving right along to Section 2, building the super plant. So, how do we actually apply these basic DNA tools in the real world to build these superhero crops? I'm talking about plants engineered to be completely immune to insects or to survive massive droughts. Well, when constructing a plant gene package, or a gene construct, as the scientists say, you need three things: a coding region, a terminator, and, crucially, a promoter. Think of the promoter as the molecular light switch for the DNA. This switch is absolutely critical because it tells the plant exactly when, where, and under what conditions to turn our newly inserted super trait on or off. Without it, the plant literally wouldn't know how to use the new gene. And getting those genetic switches right, it leads to some incredible real-world applications. Look at agriculture. We have things like Bt cotton, which is engineered for built-in pest resistance. And in medicine, it gets even wilder. Scientists are actually working on edible vaccines. Yes, using the edible parts of plants to produce immunogenic proteins. It really proves that plants aren't just crops anymore. They're transforming into these highly sophisticated living green factories. But you might be wondering, how do scientists actually get these new traits safely inside a plant in the first place? Well, meet Agrobacterium tumefaciens, nature's very own genetic engineer. In the wild, this bacteria naturally injects its DNA into plants to cause disease. But scientists cleverly disarmed its disease-causing T plasmid. Using what's called a binary vector system, they split the genetic machinery into two parts: one easy-to-handle plasmid holding our desired gene, and a helper plasmid providing the transfer machinery. It is a brilliant workaround that delivers useful genes safely and directly into the plant's nucleus. All right, let's head into Section 3, the magic plant laboratory. Now that we've engineered the perfect cell, we need to actually grow it. We're scaling up from manipulating single invisible genes to generating thousands of identical, full-grown plants from scratch. And this entire field relies on one incredibly magical foundational principle. Listen to this quote: "Cellular totipotency, the inherent capability of a living nucleated plant cell to divide, grow, and regenerate into a whole, complete plant organism." I mean, that is just amazing. It means a single microscopic piece of a leaf contains the complete blueprint and the full potential to recreate an entire jungle. To unlock that huge potential, you need the perfect environment. Scientists start with something called a callus, which is an unorganized, actively dividing mass of cells. They grow it in an absolutely sterile environment on an artificial medium, and it gets fed a perfectly balanced diet: macronutrients, micronutrients, vitamins, sugar, and most importantly, master regulator hormones. Things like auxins to stimulate root growth and cytokinins for shoot growth. Let's move to and see how this builds directly into a massive industrial process called micropropagation. First, we take our explant and stabilize it. Then, we use those hormones to force rapid shoot multiplication, induce the roots, and finally harden the new plants so they can actually survive outside the lab. This rapid commercial cloning process, it's exactly how uniform crops of bananas, orchids, and sugarcane are cultivated all over the world today. And the lab allows for even wilder experiments. By using enzymes to completely strip away the rigid cell walls of plant cells, scientists create what are called naked protoplasts. Scandalous, I know. But without those walls, scientists can literally fuse sexually incompatible plant species together. It's a process called somatic hybridization. It's just an incredible way to mix traits that nature would never, ever normally allow to cross. So with all these single-cell manipulations, genetic edits, and crazy new hybrid combinations, how do we actually manage the massive, massive amount of biological data required to engineer these future jungles?
That brings us to our final stop, Section 4. Breeding the future.
To manage all this data, we have to leave the traditional greenhouse behind and witness the marriage of supercomputers and farming. As our source material beautifully puts it, bioinformatics is the exact point where biology and computer science tie the knot. It's the application of computational tools to store, analyze, and interpret massive biological data sequences. Because when you think about it, DNA is basically just lines of code: A's, T's, G's, and C's. So we really need the immense power of computers to read and write it effectively.
Because of this, scientists globally store their findings in primary and secondary databases. Repositories like GenBank hold the raw experimental data, while PubMed holds vast amounts of research literature. It's essentially this massive shared digital DNA library that allows researchers anywhere in the world to collaborate on building better crops.
And why are we doing all this heavy computation? To support the core objectives of modern plant breeding: high yield, disease and drought resistance, better nutrition, and early maturity. I really want to emphasize this. While the technology, like CRISPR, AI, and bioinformatics, is totally brand new, the actual goals of plant breeding remain exactly the same as they were for ancient farmers thousands of years ago. We just want crops that are tougher, faster growing, and more nutritious. So the crucial point is that how a breeder approaches these goals completely depends on how the plant naturally reproduces. For self-pollinated crops, like wheat for instance, breeders use methods like pure line selection and single-seed descent to maintain pure family histories. But for cross-pollinated crops, like maize, they have to use techniques like recurrent selection and inbred line crossing just to wrangle all that genetic variability. In fact, to tightly control the breeding of cross-pollinated crops, breeders will deliberately use plants that exhibit male sterility, meaning the plant produces no pollen at all. This allows the breeder to perfectly control the entire pollination process. And the result? A hybrid offspring that exhibits heterosis, or hybrid vigor. This means the hybrid plant is significantly stronger, larger, and higher yielding than either of its parents. It's a real game changer for food production. Which brings us to this really powerful synthesis from our source text: old farming plus modern genetics equals future agriculture. Modern agriculture isn't at all about replacing nature. It's about seamlessly blending ancient agricultural wisdom with the absolute cutting edge of biotechnology and computer science. We've seen the molecular photocopy machines, the tissue culture cloning labs, and the massive digital databases. Biology just isn't a science of observation anymore. It's a programmable tool. So I leave you with this thought. If DNA is the ultimate coding language, how will you program the crops of the future to solve our greatest global crises? Thank you so much for taking this tour with me, and I really hope you leave feeling as amazed by the potential of plant science as I am.